All right, let's get into it. Today we're going on a pretty epic journey. We're going to start with the tiniest little machines buzzing around inside our cells, and we're going to scale all the way up to the complete genetic instruction manual for an entire species.
So let me ask you a question. What if you could actually read the entire instruction manual for a living thing? I mean, the whole book, not just a chapter here or there. Well, guess what? That's not science fiction anymore. That's exactly what we can do right now. Let's dive in.
So, to even begin to understand this giant instruction manual, you have to start with what it actually builds. And that means we have to start with the cell's workforce, the proteins. I mean, think about it. Proteins are the engines. They're the messengers. They're the literal beams holding the cell together. They do pretty much everything. So if we want to know what a cell is actually up to at any given moment, we need a way to see all of its proteins all at once. The first trick up our sleeve is a technique called isoelectric focusing, or IEF for short. It's pretty clever. You see, every single protein has this unique pH level where it has no electrical charge. IEF takes advantage of that, using a pH gradient to make all the proteins line up, perfectly sorted by this one specific property. But, just sorting them by one thing isn't quite enough to see everything. So here's the brilliant part. Scientists add a second dimension. They take that perfectly sorted line of proteins, turn the whole thing 90 degrees, and then zap it with another electric field. This time, it separates them all by size. And what you get is this beautiful, detailed, two-dimensional map of every protein working in that cell. And this is why that map is so incredibly powerful. What you're looking at here are two of these protein maps. On the left, you've got cells that were treated with insulin. On the right, cells that weren't. Now look closely at spots 1 and 2. See how they've shifted? That isn't just some random smudge. That is a visual readout of the cell physically changing its machinery because it got a signal. This is what proteomics looks like in action. It's amazing. Okay, so we've seen the workers. We've seen what they're doing. Now, let's go find the blueprint they were all built from. It's time to read the genome. So, when a genome sequencer reads DNA, it basically just spits out a huge text file. The old school way to do this was called a FASTA format, which just gives you the letters, A's, C's, T's, and G's. But modern sequencing uses something called the FASTQ format, and it adds something that is absolutely 100% crucial, a quality score for every single letter. And that quality score has a name. It's called a Phred score. Now, it's based on a logarithmic scale, but what it all boils down to is this. It's a number that tells you exactly how confident the sequencing machine is that it got that letter right. The higher the score, the lower the chance of a mistake. So, what's a good score? Well, the gold standard in the industry is usually what's called a Q30. A Phred score of Q30 means there's only a 1 in 1,000 chance that the base call is wrong. To put it another way, that's 99.9% accuracy. And that is the level of quality you need if you're going to try and build an entire genome from scratch. But here's the catch. We can read the letters with incredible accuracy, right? The problem is, the sequencer gives them to us in millions, and I mean millions, of tiny, shredded, overlapping pieces. So our next job is basically to solve the world's most complicated jigsaw puzzle. The whole goal of genome assembly is to build this hierarchy you see here. We start with those millions of tiny reads that come off the machine. Then, by finding where they all overlap, we start stitching them together into longer, continuous chunks of sequence called contigs. And from there, we use other clues to figure out the right order and orientation of those contigs, creating these even bigger pieces called scaffolds. So how on earth does a computer actually solve this insane puzzle? Well, one of the most powerful tools is something called a de Bruijn graph. It's a really smart approach. Instead of trying to overlap the entire long reads, the computer algorithm breaks every single read into even smaller, overlapping pieces called k-mers. And then it just has to find the simplest, most logical path through this gigantic web of connections to piece the original sequence back together. All right, so you've done all that work, you think you've assembled the genome. How do you know if you did a good job? Well, we basically give ourselves a report card, using stats like N50 and L50. Imagine you take all your finished contigs and line them up, from the longest one all the way to the shortest. The N50 is the length of the contig where you finally cross the halfway point of the entire genome size. A bigger N50 is better. It means your assembly is way more continuous and less broken up into tiny pieces. Okay, the puzzle is solved. We have it, the full sequence, our book of life. But right now, it's just a string of billions of letters. We have no idea what it says. We need to figure out what it all means. It's time for annotation.
Annotation is where we become translators. It's the whole process of going through that raw sequence of letters and adding all these layers of meaning. It's how we find where genes start and stop, how we identify all the little switches that turn them on and off, and ultimately, how we figure out what each part of the code actually does.
You can kind of think of it in two big chunks. First, there's structural annotation. That's like finding the grammar of the book, locating the genes, which are like the sentences, and finding their start and stop signals, which are basically the punctuation. Then comes functional annotation, and that's the next big step, figuring out what those sentences actually mean. So we've read the book, we've translated it, now we get to the really, really cool part. We can start putting different books of life side by side and see how they're related, how they're different, and what that tells us. And let's start with a really famous number, 98.8%. I'm sure you've heard that before. That's the percentage of DNA that we humans share with chimpanzees, our closest living relatives. That one number comes from comparing our entire genetic blueprints. And man, does it tell a profound story about evolution? And here's how we actually visualize those comparisons. This is called a syntenic dot plot. On one axis, we've got the genome of E. coli, and on the other, a related bacterium, Salmonella. Every single dot represents a similar gene between the two. And that long, clean diagonal line, that's the amazing part. It tells us that not only are the genes themselves similar, but their order along the chromosome has been kept almost perfectly intact for millions and millions of years. Now, let's see what happens when we zoom out and do this for more complex life. This incredible map compares the chromosomes of humans, chimpanzees, and mice. Every colored block is a chunk of the genome that's been saved through evolution. You can see the unbelievable similarity between humans and chimps. But you can also see that those exact same blocks are all there in the mouse genome too. They've just been shuffled around into a different order. You're literally looking at a map of our shared history as mammals. So, from watching a single cell react to insulin, to piecing together a creature's entire genetic code from scratch, to mapping our own deep evolutionary history, we can now read the blueprint of life with a precision that's just staggering. And that leaves us with one final and very profound question. Now that we have this power, what will we choose to build with it?